Hi, my name is Philip Bruckner, and I'm a PhD candidate at the Florey Department of Neuroscience and Mental Health at the University of Melbourne. My research focuses on clinical applications of advanced diffusion MRI, and I work under the supervision of Dr. Robert Smith, Dr. Remy Kamita, and Dr. David Bond. Together with our collaborators at the Medical University of Vienna, we recently published a study in Brain investigating how surgical disruption of white matter can lead to downstream atrophy in the non-resected human brain. What motivated the study is the observation that following surgical resection, there can be atrophy in parts of the brain that were never touched by the surgeon. While brain surgery generally is a very safe and effective treatment, its precision could be further improved if we could anticipate when and where such atrophy will occur. For this study, we hypothesized atrophy to occur specifically at the endpoints of disrupted white matter bundles, closely relating to the concept of transneuronal degeneration, where a surgically disrupted axon degenerates, resulting in a loss of electrical signal and nutritional support for a neuron downstream, which then in return can also deteriorate, including its projections elsewhere. While plausible, it is important to stress that most human evidence of this mechanism stems from the visual system, and it's still unclear how well it generalizes to other brain networks. Nonetheless, this was our hypothesis, and we decided to evaluate it using in vivo MRI, specifically investigating two surgical treatments for epilepsy. The first, anterior temporal lobectomy, or short ATL, is a quite extensive removal of the anterior third of the temporal lobe. And the second, selective amygdalohippocampectomy, or short SAHI, is a more targeted removal of amygdala and hippocampus. So how did we model transneuronal degeneration from neuroimaging? Well, for each patient, we first parcellated the brain into anatomical regions and then removed those portions that had been resected. For each region, we then measured three effects. First, the fraction of white matter intersecting the resection cavity, which we deemed lost to resection. Second, the change in cortical thickness from pre to post surgery. And third, the longitudinal change in connectivity to the rest of the brain, specifically focusing on those connections that were not resected during surgery. Through these effects, we then built an imaging model of transneuronal degeneration, predicting longitudinal changes based on the white matter lost to resection. To do this reliably, we developed a tailored longitudinal imaging framework that combines robust methods for structural and diffusion MRI reconstruction. We start by building unbiased within-subject templates, to which we then transform all pre- and postoperative images of a given subject. For T1-weighted images, we use the longitudinal free-surfer pipeline, which improves the accuracy of cortical thickness estimates by leveraging shared information between time points. Essentially, it reconstructs cortical anatomy only once from the unbiased template and then refines the estimate on session-specific data. For quantitative tractography, no equivalent method existed, which is why we adopted a very similar principle. For each patient, we reconstruct white matter anatomy only once from the unbiased template, but then refine the estimate on session-specific data. This approach allowed us to robustly estimate longitudinal gray and white matter features within a common output space. So what did we find? In ATL patients, we saw pronounced white matter losses, in particular in regions close to the resection. These areas also showed notable cortical thinning, and also downstream connectivity decreases were most pronounced in these regions, although strong effects were here observed across the entire ipsilateral hemisphere. For SAHI patients, we found a very similar pattern of these effects. Having now modeled our three effects of transneuronal degeneration, we proceeded to evaluate their relationships. First, we tested if a region's change in cortical thickness can be predicted based on the white matter lost to resection. We found a statistically significant relationship between the white matter lost to resection and the change in cortical thickness, with each dot representing the brain region of a participant. The model suggests the more white matter is resected during surgery, the thinner a region will become after surgery. We then asked, can white matter losses also predict changes in downstream connectivity? And I will now show you two regression plots testing this relationship. The first one will show connectivity changes that were reconstructed using standard methods, and the second one will show connectivity changes that were reconstructed using a new robust method. Using standard methods, we found no relevant relationship between the effects. Using a new robust method, however, the relationship could be modeled much more reliably, showing a statistically significant association between the white matter lost to resection and the change in downstream connectivity. This suggests the more white matter is resected during surgery, the more atrophic downstream connections will become after surgery. In combination with our first model, this indicates that the structural effect of resection propagates along anatomical networks. We think this finding to be quite exciting for a number of reasons. From a surgical perspective, it raises the prospect of predicting downstream atrophy from preoperative tractography data. From a neuroscience perspective, it provides in vivo evidence to support transneuronal degeneration across the brain's widespread anatomical networks. And from a methodological standpoint, a new method enables robust longitudinal reconstruction of white matter, opening exciting development avenues for precision medicine applications. With that, thank you very much for your interest in our article. Please feel free to share it within your professional networks, and if you have any questions regarding the analysis of methods, please don't hesitate to get in touch anytime.